Okay, now we're going to start looking at the first two steps of metabolism. First thing we're going to look at is glycolysis. Okay, the vast majority of organisms, both prokaryotes and eukaryotes, use glycolysis as part of the metabolism. Now, granted, we mentioned some oddball things that use metal oxidation, sulfur reduction or oxidation, methanogenesis and stuff like that, but those are really kind of oddball. But the common bacteria that we're used to, many archenes do this, all eukaryotic cells, from protists to plants to fungi to us, they all do this kind of stuff. Okay, now glycolysis. A couple things about it. First of all, it's an anaerobic process. It works perfectly well in the absence of oxygen. So anaerobic organisms can squeak out an existence, not a very good one, but they can squeak out an existence by glycolysis and fermentation alone. Not that's a very good way of doing it, but they can make it. Okay, second thing, it occurs in all cells in the cytoplasm. It's a series of enzymes that are in the cytoplasm. In eukaryotes, it's believed that the enzymes are loosely attached to cytoskeleton, so they're all physically close together. That way, the product of one reaction can quickly get grabbed by the next enzyme and reacted on and so on. Because it's a series of several steps. Okay, third thing. What goes in and what goes out? Well, you take one glucose and you end up with two pyruvic acids. Okay, energetic molecules. Well, it turns out you have to put in 2 ATP. You actually have to use 2 ATP to start this process. And you also have to use 2 NAD+. And what do we get on the outside? You get a total of 4 ATP, so you're up 2. Our net gain is 2 ATP, and you get 2 NADH. Now, by the way, most of the time the NADH from these processes in the cytoplasm, most of the time that is used in processes in the cytoplasm and not transported in the mitochondria. So most of the time you're not going to get too much energy out. You're not going to metabolize the NADHs for energy. You use them in the cytoplasm rather than just pumping them into the mitochondria, getting more juice out of it. Okay, so that's our output. We get a net gain of 2 ATP, 2 NADH. Okay, that's just the overall summary of glycolysis. Now let's take a look at the process. There's about 7 or 8 steps in glycolysis, and until you take biochemistry or have taken it, you're not going to be responsible for each step. But you can kind of separate this thing out into 3 kind of phases. We do 3 different things, 3 things, and some of them take a couple different steps to do so. So we're going to break it down kind of like the 3 acts of glycolysis. Now, first thing for glycolysis, the fuel for glycolysis is the 6-carbon sugar glucose. Now, most of the polysaccharides that we use for energy things like glycogen, starch, those are going to be made entirely of glucose. Some organisms can even break down cellulose, but not too many of them can do that. Okay, now the other thing is you have other 6-carbon sugars. We've got enzymes that can interconvert one 6-carbon sugar and another, and that's pretty much universal already. That's how they get that high fructose corn syrup that they put in sodas and stuff like that. What they do is take glucose, they throw some crude yeast enzymes in, turn half, corn syrup's glucose, turn half the glucose into fructose, and that way you put less in your soda, you save money, you pocket the difference, and what have you. But we can interconvert these 6-carbon sugars into glucose. All right. Now, first step in glycolysis, the idea is we're going to try to split that sugar into 2. The problem is glucose is a very stable molecule. It's not easy to split. So what we're going to have to do is modify that sugar and make it easy to split. And the way we're going to do this is we're going to attach two phosphate ions to it. Now recall, phosphate ions are negatively charged. That means they're going to repel each other. If they're right next to each other, one covalent bond apart, what's going to happen is that repulsive strength is enough to weaken that covalent bond and make the splitting a lot easier. So we're going to turn the stable 6-carbon glucose into something a lot less stable. So first thing we're going to do is, using up an ATP as a source of phosphate and energy due to the condensation reaction, we add one phosphate to the glucose. So we have glucose 6-phosphate. That P represents the phosphate. Okay, we're not done yet. We're going to convert the thing into fructose 6-phosphate. I don't know why, but... And then we're going to add a second ATP, a second phosphate, once again from ATP. This is where we're going to use up our 2 ATPs at the beginning. And now we have fructose 1,6-diphosphate. Okay. All right, now when we get to this point, here's this sugar. It's got 2 phosphates, and the number 1 and 6 carbon are very close to each other. Those phosphates are repelling each other strongly. They're not as strong, the repulsion is not as strong as the strength of the covalent bond between the carbons, but it's enough to weaken the bond quite significantly and make it easy to hydrolyze, to split. In fact, if you buy this stuff, it's fearsomely expensive. You darn well better store it in the -80-degree freezer, because if you leave it outside overnight, it's probably going to convert back into fructose in short order, and you're out a couple $100 for a gram of it. So it's unstable even at room temperature. Okay, so that's our first set of steps. What we're doing is making the sugar unstable. We're making it easy to split. Okay, now so that's part 1 here. Part 2 is where we actually split the sugar. Okay, so we got the 6-carbon sugar with a phosphate, with these phosphates here, and now we're going to split it in 2. And makes 2 copies of a molecule called PGAL, phosphoglyceraldehyde, if you're interested. You don't need to know that. But what is 3 carbons each with a phosphate on it. So what I'm going to do is I'm just going to draw it like this. Okay? So now we split this molecule in 2. Each of those 3 carbon molecules has a phosphate on it. Now, here's where we start getting the magic. It turns out that these molecules, if you transfer a phosphate ion, inorganic phosphate, you can actually do the condensation reaction, and that actually turns out that it's going to release energy. It turns out in this case, we can add inorganic phosphate. In other words, instead of using ATP as a source of phosphate, we just take an inorganic phosphate ion from solutions, always a little bit in cell fluids and stuff like that. And we can covalently attach it to this PGAL. So now we're going to get, and we don't have to use ATP this time. Excuse me? Add inorganic phosphate ions. Add inorganic phosphate ions. Okay, now what we're going to end up with is 2 3-carbon molecules, and they have a phosphate on each end, like so. And once again, your interest is called diphospho glycerate if you're really interested in that. Okay. So now these molecules, you got 2 3-carbon molecules, both of them have 2 phosphates, one in each end. And that finishes Act 2. And now part 3 is where we get the payback. We've got these molecules with covalently attached phosphates. It turns out if you hydrolyze the bond from that phosphate, you can attach it to ATP, or rather to ADP, to make ATP, and that's actually an exergonic reaction. In other words, the phosphate bond here to that carbon is more energetic than the bond from one phosphate to the next in ATP or ADP. So now we can transfer these phosphates to ADP and make ATP. We've got 4 phosphates here, so that means we can make 4 ATP molecules. So that means we get our original 2 back and we get 2 as a payoff. 2 is interesting. Okay, so that's our third thing. So in 2 steps, one step, we're going to remove one of the phosphates from each, pass it to ADP, so we'll do this here, ADP, and we make an ATP. So we pass a phosphate there, ADP here, and ATP here. Okay, so we generate our original 2 ATP back just by taking one of the phosphates off of each of those molecules. Do it, Puzzle. Okay. Now, our last step here is we repeat that, this time removing that last phosphate, passing it to ADP again, making our 2 ATP, which is our net gain. And these last 2 molecules with no phosphates, that is our final product, pyruvic acid. And now, glycolysis is over. Now, once again, the vast majority of organisms, both prokaryotes and eukaryotes, do indeed use glycolysis. One reason why glucose, sugar, is a major energy source, either directly or in the form of polysaccharides. But that's glycolysis. Now, once again, 2 ATP per glucose is not that much, but it is a start. And for some anaerobic organisms, that's enough to keep them going. It's not really a great way to live, but it does help. Okay, that's glycolysis. All right, now the next thing we want to look at, fermentation. Fermentation is an anaerobic process once again. In other words, it needs no oxygen. It occurs in the cytoplasm of all cells. Now the thing with fermentation, the idea behind this, it turns out fermentation, there's a whole bunch of different fermentation reactions. You can do fermentation reactions on things like acetic and formic acids, break them down to something. You can do fermentation reactions on pyruvic acid, which is what we're going to focus on. You can do a whole bunch of different fermentation reactions. Some fermentation reactions generate energy, others do not. But we're going to focus on those widely used fermentation reactions because this is how we get rid of pyruvic acid. We turn it into something that's less nasty. So but first let's put Okay, some fermentation reactions release energy, others don't. Now the kind we're going to look at, the fermentation of pyruvic acid, that does not release energy. In fact, it uses up an NADH in order to do this. But it does get rid of pyruvic acid. It turns it into less nasty and more easily excretable stuff. Because if you just build up pyruvic acid in your cell, that would be pretty nasty. That killed the cell in fairly short order. So instead, we're going to get rid of it and excrete the byproducts. All right. So we're going to focus on the fermentation of pyruvic acid. It takes a couple of steps. One of them uses up an NADH, passing the electrons to it. Okay. Now, we break pyruvic acid down, tear a carbon off of it, and we end up with a 2-carbon molecule. And that 2-carbon molecule can include acetic acid, vinegar. And recall, acetyl-CoA formation is very similar. The major difference is we're not going to release the acetic acid free. Instead, we're going to covalently attach it to coenzyme. But clearly, it was based on that, probably evolved from this process. Some microorganisms will take and produce lactic acid. That's how you get things like cheeses, sour cream, yogurt, butter, God forbid, buttermilk, and a whole bunch of other things. Okay? By the way, when you do a lot of sprinting and stuff, your muscles hurt after a while because one thing that happens is for a quick burst of energy, because it's a quick process, you break down the glycogen stored in your muscles, that's a polysaccharide, into glucose, run it through glycolysis and then fermentation. And it turns out that fermentation that we use builds up lactic acid. So after a while, the lactic acid builds up in your muscles. It hurts, and your muscles tired, weaken, stuff like that. So that's why, a good caliber athlete can do a 100-meter run in 10 seconds, but nobody can do a 1K run in 100 seconds. What's the record now? About 3 minutes or something like that? Okay, well, can't keep up that pace because the lactic acid buildup is too great. Eventually it gets metabolized down, but okay, so that's another thing, lactic acid. Ethanol is another one. And that's the foundation of a whole bunch of business, even in some cases acetone. So a lot of our commercial acetic acid and ethanol, that's generated by fermentation by microorganisms, yeast, or in some cases bacteria. The fermentation of things, the lactic acid, especially we start with lactose, the milk sugar, which is a glucose and a galactose. You do break it down, turn the galactose into glucose, metabolize it into pyruvic acid, and then ferment it into lactic acid, and that's how you make things like cheeses and stuff like that, and sour cream, and that's what happens when milk goes sour, and all that kind of stuff. Okay, ethanol. Of course, fermentation ethanol is a big thing. Not only are we talking about making beer, wine, what have you, but making the ethanol that's in your gas tank is another big thing. I mean, clearly the beverage industry by itself is many, many billions of dollars a year. Think of Anheuser-Busch and stuff like that, right? Okay. All right. So they're going to do that kind of stuff. And here again, different strains of yeast and somewhat different starting materials, different types of grades, different types of hops, different types of grapes and things like that are going to generate the different flavors of different kinds of beers and wine. And what counts is not only what the microbial strain does and what kind of side products it produces, but what kind of stuff you start out with, what type of grape versus another type. So you use the same microorganisms, 2 different types of grape, you get 2 different types of wines. You use the same type of grape, 2 different strains of yeast, you get 2 different wines. One might sell for $100 a bottle, another one might be poured down the sink. Okay. So, of course, the beverage alcohol industry is a big thing, but nowadays, of course, a lot of it is thinking about energy, domestically produced energy. Right now, if you go up to your gas station, you're probably going to put 10% ethanol in your gas tank. That ethanol is derived from corn, mostly due to lots of federal subsidies. Now, unfortunately, although that is domestic energy, unfortunately, it's not exactly a winner. It turns out both the energy use of the high-intensity growing of corn in this country, that uses a lot of energy. Fertilizers, tractors, trucks to take the stuff to processing plants, etc. You actually end up using up more energy than you get by producing from the production of the ethanol. In terms of carbon footprint, in other words, how much CO2 is expelled in the atmosphere, it comes out as a loser too. It would not be viable if it wasn't for heavy federal subsidies in the farming states like those, because that's their constituents are growing all the corn and stuff. So it really doesn't, and the other thing is it tends to drive up the price of a basic foodstuff, and that hurts poor people in developing nations. So, corn from ethanol, at least in the U.S. type of high-intensity mechanized agriculture, is actually a loser on all fronts, except that is domestic rather than foreign. But there are other things. Brazil, for instance, has a big thing. They can grow sugarcane in most of Brazil really easily. Sugarcane, of course, when you're taking corn into ethanol, you're just using the kernels. And the rest of the stuff goes into as waste. But sugarcane, there's a lot of sugar, sucrose, in sugarcane. So, it's easy to convert that into ethanol. You get a lot more ethanol per pound of sugarcane than you do per pound of corn stalks. So it's actually viable. It's a win-win situation on all sides. So Brazil is trying to switch as much of their domestic economy into ethanol, sugarcane-based ethanol. So a lot of their cars and stuff run on what's called E85. That's 85% ethanol, 15% gasoline. So they try to use as much of that domestically as they can. And they have large offshore oil deposits. The idea is run your own economy on sugarcane-based ethanol, and then sell the oil to China or to the U.S. or to whoever wants to pay the highest price for it, and make oodles of money that way. So, yeah, that actually works real well. Now, of course, the Holy Grail, there's lots of pilot work. Nothing is currently economically viable with the current price of oil. That may change. But the idea of taking cellulose, in other words, things like corn stalks, shredded up newspapers for that matter, toilet paper, whatever the case is, cellulose, and breaking it down and turning it into ethanol. So that way, if you did it from corn, you'd use the whole corn stalk, the cob, the whole 9 yards. You get a lot more ethanol out of it. So there's a lot of pilot work on that. Some of them involve microorganisms. The problem is, cellulose is very hard to break down. Even the organisms that can do it, if they have an alternate food source, they will not produce this massive enzyme complex required to break down cellulose. It's a very big, expensive enzyme complex to break down cellulose. If they have any other alternative, they simply won't produce it. They'll use the other alternative. Okay, so that's one thing. Another thing is by heat, chemicals, and stuff like that, partially breaking the cellulose down and then feeding it to microorganisms. So right now we got pilot studies. Of course, the Department of Energy finances some of these things. Currently, at least on a pilot scale, you can get ethanol equivalent of ethanol to about $200 a barrel oil. Currently, it's about $100 a barrel. So although it's not viable now, of course, at the price of oil with China and India competing with us to try to get a shrinking amount of oil, it might actually become viable. The price of oil goes up, which means your gas is going to run about $6 or $7 a gallon, but the price of oil goes up to $200 or $300 a barrel. This actually would be viable. And of course, the other advantage of cellulose-based ethanol production, once again, largely fermentation technology, is that you make it domestically. You don't have to buy it from a bunch of marginally stable nations that basically hate us anyway, right? The same people we buy oil from smash airplanes into buildings and kill thousands of people. Yeah, that's 19 of the 20 of those guys in 9/11 were Saudi nationals. Shows you what they really think about it. But they like their money. Okay. All right, well, so fermentation is also a very important thing. All right, well, answer that. Of Questions so far?